Hello students, welcome to Tungal online classes. So in the last video we discussed thoroughly the prokaryotic cell, and in this video we'll discuss thoroughly the eukaryotic cell. What is this eukaryotic cell? I have already explained very perfectly. The cell in which the nucleus has its definite boundary, that is nuclear membrane is present in that cell, and such kind of cell is said to be as an eukaryotic cell, right? So this is the fact what you know it better, right? So let us start. Eukaryotic, the meaning of this word itself says eu means two, karyon means nucleus. These cells have two nucleus in themselves, right? So these cells have two nucleus. These cells have two nucleus. Two nucleus in the sense that they have definite nuclear membrane in themselves, right? So this is one eukaryotic cell in which there is a definite boundary for the nucleus, that is nuclear membrane is present over here, and such kind of a cell is said to be as an eukaryotic cell. It is said to be as an eukaryotic cell. Then what are those organisms which are eukaryotic in nature, right? That is the question arises. Here some of the organisms like Protista, the organisms come under Protista, and the Mycota, Metaphyta, as well as Metazoa, right? So all these organisms, so they come under what? Eukaryotic cell. They consisting of their made up of eukaryotic cells, right? So protistans, you know it better, Mycota, you know it better. So these are nothing but all kinds of plants, all kinds of animals, right? So all these are eukaryotic in nature. These are all little higher animals as well as plants, that is higher organisms than the previous one, that is prokaryotes. Usually bacteria, mycoplasma. Already I have given examples for many of the prokaryotic cells, and these are all eukaryotic in nature, right? So this is the fact what you should know. Yes, all eukaryotic cells are not similar in their structure. So they are not similar, they are not always similar in their structure, they are somewhat different. Say for example, plant cell is there as well as animal cell is there. See the difference? Animal cell, plant cell, right? These are the two cells which are eukaryotic in nature, right? But these are having some of the differences. What kind of difference that you come across in plant cell? So there is a large vacuole is present over here in plant cell, and cell wall is present in this plant cell along with this one. So in plant cell usually what kind of unique features are seen? Plastids are seen over here. Plastids, that is chloroplast, are the kind of plastids are seen over here in case of plant cell. But in case of animal cell, the same kind of structures are not at all seen. Of course, in animal cell, small vacuoles are present, gas vacuoles, metabolic gases that are released into the spaces, right? So and they become gas vacuoles. There is a small vacuoles are absence of vacuoles. Sometimes they may be absent sometimes, right? And in animal cell there are some other vacuoles that we come across in lower organisms, that is food vacuoles. Then you come across contractile vacuole. Then you come across, so these are seen in lower organisms like protozoans, right? So these are seen out here. The unique feature of this animal cell itself is centriole. Centriole. What is centriole? What is centrosome? That difference I have gone through already. Centrioles are present in this animal cell, but the centriole is absent in case of plant cell. So this is how there is a difference in different types of eukaryotic cells. Eukaryotic cells are not at all so always similar in their structure. They are somewhat different in their structure. The example is plant cell as well as animal cell. Along with this one, these cells so have unique features, means so they have cytoskeletal system is seen over here. What is seen in these cells? Cytoskeletal system. The same system is not at all seen in case of prokaryotic cell. It is seen in case of what? Eukaryotic cell. This is also a unique feature of this cell itself. And next one. So there is a definite nucleus. We came to know there is a definite nucleus is present in this eukaryotic cell, and inside that one, what do you come across here? So that is nothing but chromatin. Chromatin network is present over here, and this chromatin network condenses to form what? Chromosomes. Chromatin network condenses to form very condensed structures, condensed thread-like structures called as chromosomes. And these chromosomes are commonly known as hereditary vehicles. Hereditary vehicles, they carry the information, that is genetical information, from one generation to another generation. This fact you know it better. In the previous class only I have covered all these things, right? So these chromosomes are mainly made up of two things. What is that? So one is the DNA, along with that one histone proteins are present over here. DNA as well as histone proteins together form. So what? These chromosomes, such kind of chromosomes are usually so 46 in number. That is 2n number of chromosomes are present in every body cell of the human being, right? So of the human being, right? So this is the fact, basic fact regarding the eukaryotic cell. And later inside the cytoplasm you will find varieties of what? Cell organelles. All those cell organelles are also membrane bound cell organelles. Majority of themselves are membrane bound cell organelles, and some of themselves are membrane-less cell organelles are also there. So all those membrane-bound cell organelles, what we come across inside this eukaryotic cell, let us study one by one. Apart from this one more thing, that is cell membrane. Cell membrane or plasma membrane is also studied thoroughly over here. So how the plasma membrane is, so what it has, so all these are the things that we study over here. And one more thing that you know it better, so plant cell has a definite boundary which is cell wall. Inner to the cell wall, what is present? Cell membrane is present. But in case of animal cell, cell membrane or the plasma membrane is the delimiting membrane or the boundary, definite boundary of the animal cell. So this is the fact what you should know regarding the fundamental things regarding the eukaryotic cell. So let us start with the cell membrane first, and later cell wall, and later all other cell organelles that we study one by one. Plasma membrane is a thin membrane, right, which makes a definite boundary around the cell of eukaryote in case of animals, and it lies immediately after the cell wall in case of plant cell, right? It is a thin membrane. So it is a thin membrane that makes definite boundary in animal cell, and it lies below the cell wall in plant cell, right? And this membrane is semi-permeable in nature. So semi-permeable in nature in the sense that it allows only the limited particles or so restricted particles to enter in and exit out. Semi-permeable membrane it is called as, right? So, and this membrane was called as cell membrane. It was called as cell membrane by Loewe. Loewe is the name of the scientist who called it as what? Cell membrane, and later it was renamed as plasma membrane by Nageli. Nageli is the name of the scientist, the one who called it as a plasma membrane for the first time. And such kind of membrane is not only makes the boundary of the cell of animal as well as plant cell, but also it makes the boundary or it makes the outer membrane of many of the cell organelles in eukaryotic cell, right? So in eukaryotic cell, it makes boundary in cell organelles also. In cell organelles also it makes the definite boundary for that. For example, in both plant as well as in the animal cell it makes the boundary. So for mitochondria, in mitochondria it is there, plastids, it is there, means chloroplast, leucoplast, chromoplast are the plastids. So around that one membrane is bound, and vacuole. The vacuole has a definite boundary in case of plant cell, and it is called as a clonoplast. Very important point to be noted over here. Clonoplast is also the plasma membrane itself, and it makes the definite boundary for some other cell organelles like lysosomes and microbodies, etc. Right? So it makes the definite boundary for all these things. Okay. So they are present. And what is the chemical composition of this plasma membrane? Let us see. Chemical composition of this plasma membrane. So according to two scientists, Grendel is the one, Garten is the second one. These are the two scientists, the one who observed the membrane of what? They observed the membrane of the RBC, right? So Garten and Grendel are the two scientists the one who observed RBC, membrane of RBC, that moment they came to know that, so the proportion of the lipids is 2 times more than RBC structures. What it shows is maximum part of the RBC membrane is made up of lipids, right? Very limited is protein. So it shows. So that's why. So mainly chemically this plasma membrane is made up of lipids, proteins, as well as very limited carbohydrate. Very limited carbohydrates. So totally, chemically, it is made up of lipids, proteins as well as carbohydrates. We came to know. But all these three are in different proportions, in different membranes of different cells, right? Some of the examples I am going to quote over here and the respective proportion of all these one, right? So let us see those proportion of different membranes. So different things in the membrane, that is lipids, proteins as well as carbohydrates in respective cells now. Yes, see this table, you will come to know, right? So membrane, here the membrane of the liver cell of mouse, right? Liver cell of mouse. It consisting of different proportion of protein, lipid as well as carbohydrates over here. So what is the proportion of proteins here? So it is 44, it is 53 and it is 3. 44% of protein is there, 53% of lipid is there, only 3% of carbohydrate is there. And second one, human erythrocyte. Human erythrocyte. That is nothing but human RBC. RBC has 52% of protein is there and 40% of lipid is there and 8% of carbohydrate is there, right? So in case of what? Human erythrocyte. And third one is outer membrane of mitochondria. Outer membrane of mitochondria. So this has 51% of protein is there, 51% of protein is there, 47% of what is that? 47% of lipid is there, only 2% of what is that? That is carbohydrate is there in this one. And the next one, fourth one. What is the fourth one? See here, the fourth one itself is the myelinated neuron. Myelinated neuron has 20% of protein is there, 80% of lipid is there, carbohydrate is totally absent over here. So these are the different proportions of what are the proteins, lipids as well as carbohydrates. That is the major chemical components of the plasma membrane. These are very important aspects to be remembered. Okay. Come to the structure of this plasma membrane. Structure. So there are many hypotheses that he explained different models of this plasma membrane previously. So first one is sandwich model and second one is Robertson's unit model of plasma membrane he explained, and the third one is Nicholson and Singer's fluid mosaic model of plasma membrane. Right? So among them, the most accepted one is fluid mosaic model of plasma membrane, that is Singer and Nicholson are the two scientists, the one who made the model of this plasma membrane and explained very perfectly regarding this one, and they proved so it is selectively permeable membrane or semi-permeable membrane. Right? So let us write the names of those models and so explain briefly. The first model was sandwich model. Sandwich model of plasma membrane. It was proposed by Daniel Lee Dawson. And the second one is unit membrane by Robertson, and third one is fluid mosaic model of plasma membrane. It is by Singer and Nicholson. Are the scientists the one who proposed their own models to explain very briefly regarding this plasma membrane, but the first two were not accepted because of some of the exceptional cases, and last one is fluid mosaic model. One is most accepted model of plasma membrane explained by Singer and Nicholson. Right? So in sandwich model, Daniel and Dawson they explains that here the lipid bilayer is sandwiched between two monolayers of the proteins, they say. Right? So see here, according to these persons, Robertson as well as Daniel and Dawson, they say that lipid bilayer. So this is considered, this one is a lipid bilayer. This lipid bilayer is sandwiched between the protein layers. Over here. See here, this is a protein layer. These are fatty acid chains over here. So this is how the model of plasma membrane explained by these scientists. Right? So but many of the exceptions were seen. So many of the mistakes were seen in these models of the plasma membrane. That's why the most accepted one is Singer and Nicholson's fluid mosaic model of plasma membrane. Then how is the fluid mosaic model of plasma membrane? So let us draw the diagram and explain the things thoroughly. Over now. Yes, come to the fluid mosaic model of plasma membrane now. Right? So this is the fluid mosaic model of plasma membrane, how it looks. So the microscopic study of this plasma membrane reveals that it is not made up of a single layer, it is made up of many layers, hence it is said to be as unit membrane. Robertson is the name of the scientist, the one who described this membrane as an unit membrane model he proposed, of course. Right? So what is this unit model of plasma membrane that he explains? Let us see now itself. Right? So why it is called as fluid mosaic model of plasma membrane? First of all, let us know this one. So as you know, this model was proposed by Singer and Nicholson. Right? First, Singer and Nicholson are the two scientists, the one who proposed this model of plasma membrane. these are the lipids and these are the proteins, right? Both together form this model of plasma membrane, hence it is said to be as mosaic of proteins as well as lipids. It's a mosaic of proteins and lipids. See here, many of the proteins are lying in the fluid of lipid bilayer. So this is said to be as a lipid bilayer, and this is fatty acid tail. These are the proteins, right? The proteins are embedded or lying in the fluid of lipid bilayer. Proteins are embedded in the fluid of lipid bilayer. Hence, this is described as. So, this model of plasma membrane is described as. What it is described as? So, the protein icebergs in the ocean of lipids. How it is described as? The protein icebergs in the ocean of the lipids, right? It is described as protein icebergs in the ocean of lipids. Protein icebergs in the ocean of lipids. Hence, it is called as fluid mosaic model of the plasma membrane. So, this is already I have told you, according to Robertson, it is a unit membrane. Why it is called as unit membrane? See here, this is one layer, this is another layer, this is third layer. Hence, it is said to be described as a trilaminar structure. Plasma membrane is described as trilaminar structure. It is described as a trilaminar structure which is made up of three layers. So first one means two lipid bilayers. You will find two lipid bilayers and one fatty acid tail layer. Alright. So see here, these two are lipid bilayers. These are forming the lipid bilayer, and the middle one is made up of fatty acid tails. These are, so that is. So see here, these are about 20 angstrom units in thickness. See here, 20 angstrom units in thickness, and the middle fatty acid layer itself is 35 angstrom units in thickness. 35 angstrom units in thickness. So 20 plus 35 angstrom units. Totally it is 75 angstrom units. The thickness of this plasma membrane is about 75 angstrom units, right? It is the thickness of plasma membrane. It is the thickness of plasma membrane. It is about 75 angstrom units. Lipid bilayer and fatty acid layer. So this is looking to be as the middle layer itself is said to be as a light layer, and these are dark layer. These two are dark in nature and this one is light in nature. So that kind of three layers that we come across in the plasma membrane, hence it is called as trilaminar structure. It is said as trilaminar structure. Then what actually it is made up of? Let us see over here. So chemically already we know this is made up of lipids. So the kind of lipids is phospholipids, right? The phospholipid bilayer that we come across, and we come across here the proteins. Here the proteins that we come across, right? These proteins are of two types. One is called as integral proteins or intrinsic proteins, and second one is peripheral or extrinsic proteins as well as intrinsic proteins are the two types of proteins that we come across over here, right? Extrinsic proteins are also known as peripheral proteins. Intrinsic proteins are also known as, right, integral proteins. They are also called as the transmembrane proteins. What they are known as? Transmembrane proteins. See here, the protein which is lying across this membrane means it is attached to both the lipid bilayer. It is completely embedded into the membrane, hence it is said to be as a transmembrane protein. Hence it is called as transmembrane protein. This is a partially embedded protein nowadays. Such kind of many partially embedded, completely embedded proteins that we come across over here in the plasma membrane. So this is more important aspect to be remembered over here. So these are the fundamental things what are present in the fluid mosaic model of plasma membrane. See here, lipids are phospholipid bilayer is present, and proteins are of two types. One is intrinsic proteins, extrinsic proteins. Right? Now it is very clear to you. And so there you come across one more thing on the outer surface. This is inner side, this is outer side. On the outer surface, you will find oligosaccharide chains, right? Oligosaccharides. So these are a kind of corn, these are a kind of sugars that we come across, that is, oligosaccharide chain it is. And these oligosaccharide chain that makes the carbohydrate. Already I have told you this plasma membrane is mainly made up of proteins, lipids as well as carbohydrates. That's why these are the three chemical components that we come across over here itself, right? So I hope it is very clear to you now. Okay, so let's come to the point. What is that? So these are lipids. What we come across, the phospholipids have polar head. This is said to be as polar head part, and this is called as non-polar tail part. It is said to as non-polar tail part. So this is the head part and this is tail part. So these are tails are made up of fatty acid tails. So the tails are made up of fatty acid tails, and these tails are bonded with each other by van der Waals forces, right? So they are bonded with each other. That is, the fatty acid tails are bonded with the help of van der Waals forces. Van der Waals forces, they are held together with, right? One thing. right? And next one, these proteins, extrinsic proteins, are attached to the phospholipids, and these proteins are also attached to the phospholipids, right? So, with the help of weak electrostatic forces, so the proteins attached to lipids as well as fatty acids with the help of electrostatic forces, with the help of weak electrostatic forces, right? So these points will be noted over here very clearly. Okay. So see here, lipid bilayer is there, fatty acid tail is there. Lipid bilayer, the polar head region it is, and this is non-polar tail region. So the non-polar tails are facing each other towards the center, and the polar heads are facing towards the surfaces, free surfaces, right? So this is the fact what you should remember over here itself. And see these proteins can move laterally across this lipid bilayers, right? So these lipids are enabled these proteins to move laterally, and this property itself is called as the fluidity of the plasma membrane. What it is called as fluidity of plasma membrane. Fluidity is nothing but lipids enable the proteins to move laterally across the membrane. So these proteins can move here like this across this plasma membrane. They can move themselves very easily. This is said to be as the fluidity. The property is said to be as fluidity. So fluidity depends on the percentage of lipids as well as the temperature. These two factors plays very important in maintenance of the fluidity in the membrane, right? So the fluidity depends on the percentage of lipids as well as the temperature. When the temperature increased temperature leads to a lesser fluidity, right? And decreased temperature leads to more fluidity, right? Leads to more fluidity in the plasma membrane. This fact is also very important point, right? So see here, so this is about the fluidity of this plasma membrane. Now we came to know, so actually what are the things present in the plasma membrane and how do they act themselves. It is just like the fluid. The lipid bilayer act as the fluid. In that fluid, the movement of what takes place, the integral proteins or the intrinsic proteins as well as extrinsic proteins take place. Hence, so they have already described this fluid mosaic model of plasma membrane as the protein icebergs in the ocean of lipids, right? So this is more important aspect to be remembered over here. Okay, come to the next important point. See here, there are two types of proteins that we come across. One is intrinsic proteins and the other one is extrinsic proteins. See here, So I shall write here extrinsic proteins which are lying on either surfaces. Okay. So here these extrinsic proteins are present or provided on either surfaces of the membrane. This is inner surface, this is outer surface, remember. And these are extrinsic proteins and these are said as intrinsic proteins. Intrinsic proteins. Intrinsic proteins are also called as transmembrane proteins, right? So why? Because they are lying across, vertically across the membrane. Hence, it is said to be as what? Transmembrane proteins. These are called as. Is that very clear? And such kind of intrinsic proteins sometimes singly means only one intrinsic protein may be present, or in chain, right? Or in chain, they provide a channel for the movement of particles from outside to inside, from inside to outside, right? So these are acting as the channels. These are acting as the channels for the movement of molecules across the membrane, right? So this is more important aspect we remember regarding the intrinsic proteins. Okay, yes. And one more point. To these intrinsic protein or extrinsic protein, to these proteins as well as lipids, so there the oligosaccharide chains are attached over here, and they form, if they are attached, oligosaccharides are attached to the protein, they form glycoproteins. See here, I shall write here glycoproteins. And so if they are attached to, means oligosaccharides attached to the proteins, then they are called as glycoproteins. So or else glycolipids, they form glycolipids if oligosaccharides bind into the lipid system. This is called as what it is called as glycolipid, right? And these oligosaccharides, they act as the surface recognizers, right? So the surface recognizers, why they are surface recognizers, they are acting as, right? Why they are called so? Because these can identify self as well as non-self of the body. Means they can so identify whether it is the property of our body or whether it is the part of our body or not. Means many of the times what happens, so to this cell there is a possibility of entry of the antigens or the pathogens. And the pathogens are foreign bodies, they are not the self of the body. Means they are not confined to the body itself, right? That's why pathogens, if they come in contact with these oligosaccharides, they recognize them as the foreign body. Means these are the oligosaccharides which can recognize self things as well as non-self things of the body, right? So this is more important point related with NEET examination. Alright, so this is about what? Oligosaccharides. That is, they form glycoproteins if they are attached to the proteins. They form glycolipids if they are attached to the lipid bilayer itself. Right? So like this, majority of the time they are towards the outer surface, and this is all about the Singer and Nicholson's fluid mosaic model of plasma membrane itself, right? I hope you have understood the entire structure itself. So how to explain that one? So briefly I will explain once again over here. So simply It's a trilaminar structure which is made up of 3 laminas or 3 layers. 2 are lipid bilayers. One is fatty acid tail. So the central one is lighter one, and both the lipid bilayers are darker in nature. And totally the thickness of this lipid layer itself is 20 angstroms, and this is also 20, and this is 35 angstroms. Totally the thickness of this trilaminar structure is 75 angstroms. Okay, and such kind of the trilaminar structure of plasma membrane that contains so lipids, that is phospholipids, and the proteins, that is two types of proteins. One is extrinsic proteins and another one is intrinsic proteins. Intrinsic proteins are called as integral proteins, or sometimes they are vertically arranged across the membrane itself. So then they are called as transmembrane proteins. And another one is extrinsic proteins. They are arranged on either surfaces of the plasma membrane, and these proteins act as the channels, ion channels, because they have the movement of the ions, so on either surfaces of, so means across this membrane itself, right? Integral proteins. And one more thing is the fluidity, the property called as fluidity. So here in the lipid fluid itself, these proteins are enabled to move across this membrane, and that is called as fluidity. Fluidity depends on the number of lipids, that is percentage of lipids, as well as the temperature. If the temperature is high, fluidity is less. If the temperature is low, then fluidity is more. So fluidity, if it is more, then easily the proteins can so move across this membrane very easily. Alright, and one more thing, if the oligosaccharides, if they are attached to the proteins, they form glycoproteins, and if they attach to the lipids, the glycolipids they forms, and these are the surface recognizers. They are acting as, they can recognize, or they are called as recognizing markers. They are acting as, and they recognize self as well as foreign bodies which come in contact with the plasma membrane over here, right? So this is all about the plasma membrane, that is fluid mosaic of plasma membrane. So let us see the functions of this plasma membrane.